Hello and welcome to the third session of the SimScale Biomedical Engineering Workshop. It's great to have you all here today with me. My name's Anna and I will be presenting this third and final session on in vivo blood flow. I think I introduced myself in the first session, so just a little bit about me. I'm the Academic Program Manager here at SimScale. And let's get right into today's agenda. So we're going to talk today about in vivo blood flow. So this is basically blood flowing through any living organism. In this case, we're going to talk about the human body. And so that's kind of the first thing that we're going to discuss. We're going to discuss blood circulation and viscosity when it comes to blood. Then we'll do a live demonstration with a simulation exercise. This time we'll focus on CFD instead of FEA like we have done in the past two sessions. So we're going to kind of switch focus. And third will be the key learnings. Then I will hand off a homework assignment to you that you guys have a week to do. So you'll have until the 18th to submit all three homework assignments if you haven't submitted the first two yet. But that third assignment will be due on the 18th, and then we will wrap this final session up with a question and answer session. So to introduce things, let's talk about blood circulation. So basically the heart is divided into two sections. We have the left atrium and the right atrium. And in this diagram on the left, you can see the oxygen-rich blood is depicted in red. Oxygen-poor blood is depicted in blue. And what happens is the oxygen-rich blood is leaving the left atrium of the heart and traveling through the largest artery first, the aorta, and then down and spreading through the body through ever branching smaller and smaller arteries down to capillaries. And this is how your body is getting oxygen. All the cells in your body are getting the nutrition that they need. Once that oxygen is depleted and the nutrients are depleted, that blood flows back through veins into the right atrium of the heart. So that's the blue oxygen poor blood that you see entering the right atrium of the heart. From there, that blood is sent to the lungs where it receives oxygen. And then that blood is again transferred into the left atrium of the heart, and the heart is acting as a pump. So it's taking this oxygen-rich blood and pumping it out through the aorta into the rest of the body. And so we'll see the highest pressures in the aorta region. And we can talk about next the three different vessel types that we see in this system, this circulation system. The first being the artery. And so arteries have a mean diameter of about 4 millimeters and a thickness of 1 millimeter. So you can see with the comparison that they have relatively thick walls. And we can ask ourselves why. I think the reason here is because we have the highest pressure. So when the blood is being pumped out of the heart, there's high pressures. And so these arteries need to have strong walls to withstand those pressures that are being pushed onto it. The next would be, we can talk about veins. So veins are what's bringing the deoxygenated blood back to the heart. These are also quite large, 5 millimeters in diameter, but not so thick. And this is because, again, the veins are bringing back oxygen-poor blood and the pressure is not so high as the blood is being drawn back into the heart. And so the final vessel type we can talk about are the capillaries, and these are the smallest blood vessels. And capillaries have very, very thin walls, actually. So, and they're quite small anyway. So the mean diameter is 8 micrometers, and the thickness of the wall is 0.5 micrometers. And capillaries have very thin walls because they want to allow oxygen and other nutrients to pass easily in and out quickly. And this is a process that's called diffusion. So it's basically a different process than what we're seeing in the arteries and veins, which are just, we're having really blood flowing through arteries and veins, where in capillaries, they're quite small and the blood is just kind of diffusing through the cells. And we can have a closer look here about the pressures that I was talking about. So again, as the blood is leaving the heart, this is where we're seeing the highest pressures. So the aorta and the large arteries are experiencing the highest pressures as that blood is being pushed, the fresh blood is being pushed out of the left atrium, and then that pressure is going to keep decreasing from into the capillaries and then all the way into the largest veins, where it's quite low as the blood is re-entering the heart through the right atrium. So the next thing that we can talk about is a concept called viscosity. And it's a very important property of blood. So viscosity, if we look at the kind of mathematical definition that you can see at the bottom, is a ratio of shearing stress to the velocity gradient. And this is a little bit hard to understand, I think. So we can also think of it as blood's resistance to flow. And if we know Newtonian fluids, such as air and water, the resulting shear of the fluid is directly proportional to the force that's applied. And this is a linear relationship, as you can see on the right side of the screen with this blue Newtonian line. So the shear rate, the relationship between the shear rate and the stress is linear. Blood is a little bit more complicated of a fluid. It's not really similar to air or water, obviously, but it's a non-Newtonian fluid. So the viscosity varies, and it varies dynamically during each cardiac cycle. So depending on where the blood is in your body, if it's in an artery or vein or capillary, it's going to have a different viscosity and behave differently. And if we look a little bit closely here, we can talk first about what blood is made out of. So it's not really a homogeneous substance. It's basically 55% plasma, which is plasma is primarily water, 91% water, with small additions of proteins and other solutes. And then we have 45% of blood is made of formed elements, and so these are what we think of when we have platelets, which are involved in clotting blood, white blood cells, which you know about because they help your immune system, and then red blood cells, which are the cells that are actually carrying oxygen and helping remove CO2 from your body. And if we can see on the left that this is even shown as being separated. If we put blood in a centrifuge, for example, we can completely separate the plasma from the formed elements, and we can distinctly see that they're separated substances. In your body, they're mixed together. Plasma is kind of a medium that's holding, the blood cells are basically suspended in plasma when it's moving through your body. And now we can move on to what the viscosity of blood depends on three things: the shear rate, vessel diameter, and hematocrit. So let's talk about these more in detail. The first is the shear rate. So blood viscosity, higher blood viscosities are associated with lower shear rates, and this means these are going to be thicker flows. A higher shear rate means the fluid is moving quicker and more easily through the body. The second thing is the vessel diameter. And so we can see here, we're talking about apparent viscosity, but basically with larger tube diameters, all the way down to about 8 micrometers, the viscosity is decreasing. And this is for a number of reasons, but especially as the tube diameter gets down to about 10 to 20 micrometers, the formed elements are more concentrated in the center of the tube, while the plasma kind of goes to the outside. and acts as lubrication so the flow can move more easily. When we hit about 8 micrometers, this is the size of a red blood cell. And so that's why you can see the viscosity is going to infinity at about 8 micrometers, because this is where basically the red blood cell is going to get stuck. And so the viscosity is going to become extremely high because the red blood cell can no longer move. And the final thing that's affecting blood viscosity is the hematocrit. And this is a bit of a strange word, but basically all it is it's referring to the number of red blood cells as a part of the total volume of the blood. So the more red blood cells that you have in the blood, the more viscous the blood is going to be. And so that was just a nice short intro to the cardiovascular system, your arteries, veins, how blood viscosity affects what's going on in the body. And so now we can switch gears over to the simulation that we're going to do today. As I said, in this session, we're switching from, in the previous 2 sessions, we did FEA, more FEA-focused exercises, and today we're going to look into doing a CFD, computational fluid dynamics, simulation of blood flow through an artery and an artery bifurcation, so where the artery is splitting. And so if we look at the setup, it's quite simple. We basically just have an inlet and 2 outlets. And the pressure difference in this case, we're taking from the flow rate through a healthy artery. So we have an inlet, a higher inlet pressure and lower outlet pressures, because we know fluid moves from high pressures to low pressures. And so this pressure is taken from a known artery case. And so that's basically the introduction, and I'm going to switch to the SimScale platform now so we can go through how to set this up. And this is what you're going to be looking at in your homework today. You're going to be able to look at, we have 3 arteries. The first one being a normal artery, so we can see both, we have full flow in both of these branches. In case 2, we have a blockage. So what a blockage would look like, it would reduce the flow by 60% in this area. And so this is case 2. And case 3 is an 85% blockage. And so you can see a very, very actually thin amount of fluid is coming through this artery. So we're going to look at how that affects the velocity, the pressure in the artery, and also the viscosity of the blood in the artery with these 3 different conditions. So the first thing to do is create the mesh. Let's just look at one case. We can look at the 85% case, how to set up this mesh. And so the first part, we can look at the operation first. This is a hex dominant parametric mesh. We use parametric meshes to give us more control over the meshing process. And so in this case, we've meshed this by creating a bounding box. So we tell SimScale that this is the area that we want to mesh, and then we use the material point, which is located inside the artery, to help the meshing algorithm know that we want to mesh the inside of this volume that's inside the bounding box. And then another thing that we've added here is a refinement. And I can zoom in so you can see this better. This small cylinder we've added as a geometry primitive to help us refine this very thin section. We need to have smaller cells here to help us resolve the fluid flow. And so that's the basic premise, and we're using a hex dominant mesh, and we've added a number of refinements for the layer. We've refined the entire surface. We've added layers, and so this actually helps us resolve the fluid flow. If I zoom in closely, you can see these layers. These very thin layers near the edge. We've added that. A feature refinement, and then here we have the additional refinement at the cylinder, which is for the very thin part of the artery. And so that's what our final mesh looks like. And we set up the mesh for each of the 3 cases, so case 1, 2, and 3. You'll do this in your homework, so you'll get to go through the steps very much in detail. So I'm just going to give you kind of an overview right now. In the simulation designer, the first thing to set up is this is going to be an incompressible flow. This is the analysis type. And when we pick the analysis type, it's going to expand the tree, which gives us these items that need to be filled in. Right now, everything has been filled in because this project is complete, but as you're setting things up, you'll see anything that has a red circle by it needs to be filled in as you're working through the project, and this helps us go through step by step. So we're using an incompressible model. Our turbulence model is K omega SST. This is a very standard turbulence model. So we know that there's probably going to be some turbulence with the way that this artery is set up. And then steady state, simple solver, these are set by default. The next thing that we need to do is set up the material. And as we discussed, blood is not a we need a viscosity model when we're modeling blood. It's not a Newtonian fluid, so we don't have that linear relationship, and we need to provide these additional factors. We're using a model, the Bird-Carriot model, to model the blood, and we can see that blood has a very similar density to water in this case, but the flow parameters are quite different than what we would expect with water. And we assigned the blood to the entire volume of the artery because that's what we're modeling. We're modeling the blood flow through this artery. Next, we have the boundary conditions. So this is what's helping is giving the system its boundaries, telling the solver how the fluid should move. And so at the inlet, we have the high pressure, so 1,184 pascals at the inlet here. The outlets have a gauge pressure of 0. So this is giving us a pressure gradient through this artery, as we would expect to see. Areas of high pressure to low pressure, the fluid moving, and then the artery walls. And these are set to a standard no-slip velocity treatment. So that's the basic setup in SimScale. It's a pretty simple setup, and then we will run this flow. And we can set, one last thing to discuss is this result control. We can set an area integral, and this will allow us to see the mass flow through the artery. So right here you can see we've already run all three setups, and now we can go in and compare the results. All right. So the first way we can do this is we can actually compare, I'm on the 85% blockage still. We can compare all three by using the transform function as we did in the first homework. So you guys should be familiar with this. And we can use that to do a comparison of especially the flow velocity and the viscosity. So in biomedical engineering, understanding blood viscosity is highly important, and it's been found in a number of research studies that patients that have high blood viscosity are more at risk for heart attacks and things like this. So it's an important phenomena to understand. And so let's have a closer look by viewing the results from the simulation. And so the way that we can do this is, right now we can't see much because the velocity at the walls is always going to be 0. This is the conditions that we're using with our wall conditions. But if we take a slice and we need to change the settings of the slice. And this will give us a view of the interior of the artery. One second, I think I made a mistake there. Okay, so there we go. There's a few spots missing, but that's okay. It's hard to get a perfect slice. And then another thing we can do is if we want to see the bounds of the artery, if I click on run 1, I can set this to a solid color. And change the opacity to about 0.3, which will allow me to see the outer bounds of the artery with the slice in the middle. And right now we're looking at pressure. So again, as we expect a higher pressure at the inlet and lower pressures at the outlets. Okay. So now let's do, this was the 85%, we can switch and do the 60% by right-clicking and adding the results to the viewer. And again, in this case, to move it so they're not stacked on top of each other, we use the transform filter. And so I can just move this about 0.05 meters, and we should be able to see both stacked on, or both separated. There we go. And then again, I'm going to change this to a solid. And use the slice filter to see the fluid flow. Okay. And this should give us the 65. In this case, the pressures don't look too much different, a little bit, but not too much. We'll notice bigger differences when we get to the velocity and the viscosity. And let me just do the last one here really quick. And this time we'll move it about 0.1. And one more slice, and we'll be ready to do a comparison. Okay, so now we have all three, it looks like, and right now again we're looking at pressure. Let's switch this over and look at something that's a little more interesting, such as velocity. And I have to change that for each slice. Okay, there, and we can scale it. And so we can see, as we would expect, especially with this very clogged artery, most of the fluid flow cannot, it's really slowing the velocity of the blood in this clogged section, and you're seeing higher speeds out the clear branch. So here, yeah, we can really notice the differences in the velocity flows. And the final thing we can look at in terms of this is the viscosity. And so let me again change this. Viscosity is denoted by nu. And let me scale that again. I might need to scale it with the third one. Let's see. There we go. So now we can definitely see differences in blood viscosity as well. So here we're getting high viscosities where the artery is very thinned. Here we can also see some, but not nearly as bad. And then here we have clean flow through both sides, and this is the normal artery. Okay. And so these more viscous flows are thicker. They're going to cause more pressure on the heart, and this is a phenomenon that can lead to heart attacks, for example, and all sorts of bad things happening in the body, a lot of stress. And the final thing that we can look at in terms of the results is we've also mapped the area integrals. And so this is over the inlet. And this allows us in the y direction to see the flow rate through the artery. So we would expect that a normal artery would have a higher flow rate compared to a blocked artery. And it's a little bit hard to see it on this graph, but if we go to a comparison, which I've put in the slides, we can have a look. So this kind of shows what we would expect to see because the normal, it's easier for the blood to pass through the normal artery compared to this 85% blocked artery. And we can see, based on the flow rates, the volumetric flow rate, how much can move through each of these. So that kind of, let's just do a quick wrap up of what we did. And again, you're going to go through these in in your homework, so you'll have a chance to investigate this yourself and see maybe you have some other findings or interesting things that you can write in the forum if you go through the post-processing yourself. So the first thing we did in this project was create the mesh. We uploaded 3 geometries, the normal artery, the 60% blocked artery, and the 85% blocked artery. We created a mesh using the hex dominant parametric mesher. This gives us a little bit more control over where we add refinements and the overall meshing altogether. Then we did the simulation setup. We assigned the mesh. We defined the material as blood. We used a specific model to do the blood, since we know that it's not a Newtonian fluid. We set up our boundary conditions. Our inlet was a pressure boundary condition, and our outlet was also a pressure boundary condition. And then we ran the simulation. And finally, we went through the post-processing steps. I showed you some things with the slices, the opacity, and we looked at velocity and nu for viscosity, and saw that there were major differences between the blocked artery and the healthy artery. And so the final thing I have for you guys today is the homework. And this is the third and final homework. Again, it's due on the 18th of February. So you guys should have the hang of how the homeworks go by now. You're going to simulate a healthy artery, a 60% occlusion, and an 85% occlusion, which we kind of just went through. You'll go through the steps in detail and set this up yourself. And analyze the results. And everything is available on our website at simscale.com/biomedical workshop. And I will send an email out tomorrow with this recording, as well as a link to the homework and where you can submit the homework. And so that concludes the session. And let me see if there are any questions. Okay, so Banu has a question. And her question is fully developed flow being defined by the codes in SimScale? And I mean, generally here, as we saw in this case, it's not really relevant because we used pressure inlets and outlets. If we're assigning a velocity inlet, generally we're using a fixed velocity inlet, which is not accurate. So the best practice here is to always, if we're interested in what the flow is doing here at the bifurcation and down here, then we need to make sure this outlet is extended enough that we can put a uniform velocity in at the inlet, and then the flow will develop along here. So by the time it reaches the area of interest, we have fully developed flow. So in this case, it should be totally fine. We have a long enough inlet. You just wouldn't want to, cut the inlet and make it very, very short. We have to give it a chance for the flow to develop. Okay, are there any other questions coming in today? If you have others, you can always send them to me as well. I'll show the email on the final slide, academic@simscale.com. So if you have any other questions related to this workshop or academic things in general, please just email this and we will get back to you on that. Okay, I think that's it for today. Thank you guys for your time, and finish up the homework, and hopefully I will see you again at another one of our workshops. Bye.